Good morning. I'm David Geiser, from Department of Plant Pathology and Environmental Microbiology. It's my great pleasure to introduce a PhD student, finishing PhD student. Can we use that term? Yeah. Terry Torres Cruz, and she's going to talk about aspects of her dissertation work. Terry comes from Costa Rica, and she comes to Penn State from doing a master's at Western Illinois University with Andrea Floral. And there was kind of a microbiome connection there because one of the big things that came out of Terry's master's research was characterizing a very common fungal sequence that was commonly found in soil microbiome studies all over the world, but nobody knew what the fungus was. And so Terry worked with a group that actually cultured the fungus and described it as a new genus, probably deserves a term. So she had that kind of experience going in. When it came to her project, she expressed a really strong interest in doing this microbiome stuff. And I thought, well, that could work. Wasn't really sure where it was going to go. And there was some risk to it. And all I can say is I'm really thankful to the dog group and the Microbiome Center for really making that happen. Because now that we got the data, I went from thinking, well, maybe we can get a paper out of this to how many papers maybe we can get out of this. So I'm really grateful to this group for helping us out with that, because it's the kind of thing that I personally could not have completed my work. So we really needed you for this. With no further ado, though, let me introduce Terry so she can talk about it.
Right. Thanks for the introduction, Dave. I'm Terry Torres. I'm a PhD candidate in the Department of Plant Pathology. I'm doing a dual title in Plant Pathology and Biogeochemistry. So my advisors are David Geiser and my co-advisor is Dr. Bruns. And I'll be telling you a little bit today about my dissertation project, in which our lab focuses on working with a specific genus of fungi, which is Fusarium. It's an important plant pathogen, very known for being a bad thing, but it can also do other things, which is pretty cool, and what we're gonna talk about today. And I'll be talking more on the side of the interactions of this fungus and the plant with other microorganisms and with insects in a potential new mimicry system. Okay, so we'll start by going over few definitions and examples of floral mimicry in fungi specifically. Then we'll move on to talking about our specific system, which are the Fusarium pseudoflowers. Then I'll introduce the different objectives of my dissertation, and we're actually not going to go over all of the results, but just a couple of things in terms of the interactions with insects, and then what you're all waiting for to hear about today, the microbiome story for this system. Okay, so I'm sure you're all familiar with the term mimicry. So we're just going to define it for our purposes today. So in mimicry, we have a model organism, which is going to be our species number one. And over here, it's going to be, in our case, just a flower of a plant. Then we have a second organism called a mimic. And in this case, it's going to be that fungal pseudoflower. And then there's a third organism involved. That's going to be the operator. And in a lot of these cases of flowers and pseudoflower mimicry, it's going to be a pollinator or some kind of insect that comes by, and it may be tricked into thinking that that fungal pseudoflower could be the actual flower of that model organism. So for the purposes of this presentation, whenever I say pseudoflower, I'm just referring to a flower-like structure that is induced by a fungus. Okay. So one example of a fungal pseudoflower is Volatile, which is a plant that gets infected by Coccidia monoica, which is a rust, and it'll produce this structure here. And what the fungus does is it'll infect the plant, it'll induce the plant into producing or arranging leaves into this like rosette-like structure. And then the fungus will produce spores that are that yellow pigmentation that you see on top of those leaves. Now, if we see an uninfected Botrya sticta, it looks like this one. So if you look on this picture, we have some of those leaves. As you can see, just naturally occurring Botrya sticta does not produce that rosette structure. And the flowers are actually not yellow, but are like this pink purplish color. So you're probably wondering, what is it mimicking if it doesn't look like the host plant, right? So in this case, the Puccinia monoica is actually mimicking a co-blooming plant that is present in that same area or group of plants. So Inanculus inamoenus is an example of them, but it's like the group called commonly as the buttercups, which are also yellow, like the pseudoflowers. Okay, so if we put this information back into that diagram that we saw at the beginning, our model organism will be the buttercup. Our mimic organism will be the fungus that's infecting Botrya. And our operator are going to be those insects that come and visit. And in this case, are different bees, butterflies, and flies that come by. Now, a lot of this work, or basically all of the work that has been done in this system of Puccinia monoica and Botrya sticta has been done by Dr. Biddy Roy, who works at the University of Oregon. So I'll be showing some of her results over here. So one of the things that she looked at was whether there are differences in the sugar content between those structures. So actually the pseudoflowers produce just as much or more sugar content than the true flowers of what it's mimicking. And if we look at insect visitation, they're visited just as much pseudoflowers or the real flowers. However, the insects will stay for a longer time on the pseudoflowers than they do on the flowers. It's interesting. So thinking of that, she was like, okay, well, what if the pseudoflowers were on their own? Would they still get visited? So she had 3 different plots where she had one plot was just the pseudoflowers, one plot just the buttercups, and a mixed plot. And what she saw is that actually when you have pseudoflowers and buttercups at the same time, you'll get greater visitation from the insects when they're associated with each other. And to try to explain that, she started looking at the volatile composition that of what was being produced by these different tissues. So in just vegetative tissue of the host plant, we found that it's mostly fatty acid derived products that are produced. All those 3 are examples of different buttercups, so co-blooming true flowers, which mostly produce terpenoids and some aliphatic GLVs. And then our pseudoflower produces mostly aromatic compounds. And all of this include different insect attractants. So what she found is like they're all producing different volatiles. So it makes sense that they'll bring in different insects that may come by. So when they're together, they bring in a more diversity and more quantity of different insects. Okay, and just like in that system, we have a few other examples of fungi that can produce pseudoflowers, or if we stretch the definition of pseudoflower a little bit, we can include a few other examples, like thinking of like mummyberry disease. It doesn't actually produce a floral structure, but it produces sugary rewards for insects and volatiles that would attract. So maybe if we think of like terms of function, actually functioning kind of like a flower. Then we have things like anthers moth, which it doesn't replicate the full structure of the flower, but what Microbotrium does is that on the anthers, it'll stop the pollen from being produced, and it'll produce its own spores on top. So it's actually just mimicking a part of that floral structure. However, there's only, you can see, a handful of different examples in this of fungi that can do this. So there's still a lot of questions regarding fungal, pseudoflower, floral, mimicry, and fungi. Things like the genetic basis of pseudoflower formation, variation in insect attraction, molecular regulation during that relationship, and the diversity and abundance of pseudoflower inducing fungi. You can see we have scarce documentation. Those systems have been like pretty well characterized, but if we think of the amount of fungi out there, different fungal species, and then think of like there's a handful that can do this, there's probably more fungi out there that can do something similar that maybe we're just not capturing at the moment. And that takes us to our specific system. So this person here on the left is Dr. Kenneth Wardak. He works at the Smithsonian Institution, and he was doing some botanical work in Guyana in 2012 when he was collecting different groups of plants that they were going to bring back to the herbarium of the Smithsonian. And one of those that he was collecting was the genus Xyris. They're commonly known as yellow-eyed grasses. So, it's a grass, but this high at the most. Some are taller than others. And they have this little flowers or inflorescences that they'll produce, which is why they're called yellow-eyed grasses. So he's out there collecting different species within this genus. And when you're doing that kind of work, you want to find a specimen that is representative of the area that you're sampling. So things that are odd, you probably don't want to pick for this specific purpose. But he was finding things that look like this on the right. And, he had heard about the Fucsinia monoica system, and he thought, that kind of looks similar to that, so maybe it's a rust. And he brought some samples back to try to figure out what it was. Now, we look at the distribution of Xyris species across the world. There are over 400 different species. Most of them are in the tropical regions, but some of them can live in temperate areas. All the countries here highlighted in green or blue or yellow are all countries that may have Xyris species in certain areas. Now, if we think of the Xyris species that we know produce pseudoflowers, we know of 3. So Cetigera, Surinamensis, and Bicephala. Bicephala was only observed once, and we have no samples of that. So could have been just by chance, but yeah. But those 3 host species are endemic to northern South America. So all of them are found in this area, and actually a little bit of Brazil on the north part as well. I just couldn't put it on the map as well. And the presence of the pseudoflowers has only been reported in Guyana. That doesn't mean it's not present in the other areas where the hosts are. We just don't have any reports. And that may be because people are not interested in this plant, or maybe they're located in areas that are not as accessible to like researchers. Okay, so eventually we figured out that it was not a rust. It's actually a Fusarium species that produces that tissue, and not similar to Fucsinia monoica. Instead of making like producing spores on top of the structure, that full yellow part that you see at the top is all just like mycelium. It's all fungal tissue that's produced. And we found out that and it was described as a novel species, so it's called Fusarium xyrophilum. It's a new species within the African clade of the Fusarium fujikori species complex. So Fusarium has a lot of hundreds of different species. They are divided into species complexes, and this is actually one of the complexes that it's really heavily studied. A lot of like important plant pathogens. It Some of the strains in there can produce fumonisin mycotoxins, so it's like very heavily studied. And to find something or new species within that group that can do something this peculiar, is really interesting to us. On top of that, it was a little bit hard at the beginning to figure out that it was a Fusarium species, because when you look at it in culture, it doesn't really look like a typical Fusarium. So when you think of Fusarium, people usually think of Macroponidia, like this one, so this banana-shaped spores, and this is what they usually look like in that Fujikura species complex. But xyrophilum does not produce Macroponidia, or not that we've seen. It actually produces this Microorganisms that are like slightly elongated. So overall, it's a very interesting novel fungus that we had very little information on. And so this is where my dissertation work comes in. And what I'm doing is looking at different aspects within this system to try to better describe what we're seeing. So I'm doing things like characterizing the presence of the pseudoflowers. Are there any symptoms? Are there other signs? What is the incidence of pseudoflowers? How is it distributed? I'm also looking at how it might be dispersing, or is it present all throughout the plant or just in a certain area? Can that help us figure out how it's moving around from plant to plant? I'm also looking at, in other fungal pseudoflower systems, there are insects involved. We're interested in knowing if that could be the case in this system as well. So we're looking at collecting volatiles to figure out there are different compounds produced by the fungus, or if it's mimicking those from the plant. And we're also looking at insect visitation in comparison between pseudoflowers and inflorescences. I'm also looking at what you're all here to hear about, the microbiome in both the pseudoflowers and the flowers or inflorescences, to see if there are, what can we see there? Who else is involved? And at the beginning we thought it could be a rust just because of what it looked like. Fusarium, when we think of it, we wouldn't think of it looking like that structure. So perhaps there could be other organisms involved in that interaction. But for the purposes of the talk today, I'm just going to focus on objectives 3 and 4 and show you a little bit of results in that area. Okay, so I have 3 study sites, or 4 technically, in Guyana. In region 4, which are the 2 dots here at the top, are sites number 1, this one, and number 2. And those are sites that have Xyris surinamensis, so the host for that fungus, and we see pseudoflowers and inflorescences at that spot. Then we have the Iwokrama forest, which we couldn't find any pseudoflowers in that area, but we do have Xyris species that are not the host, the known host. And then we have a site in region 10, which again, we could find Xyris, just not the host species, the known host species. Okay, so this project has a lot of different challenges. One of them is that we know very little about the system, starting from like the plant. Like, when does it flower? We don't know. Are there insects that come? We don't know. And just like that, I could tell you a lot of different questions that we just don't know about this plant. There is a Xyris monograph, and there's some information of insects there. So what it says is that there are no nectaries that have been found in any of the species in the genus. And only a few insects have been observed on the flowers. That includes some pollen gathering bees, but even those are very infrequent. And if we look at the reports that have been published regarding insects on Xyris, most of them have been done on temperate Xyris in North America. So what has been reported there are different alicted bees, syrphid flies, and some robber flies. And there's also a moth in the genus Volatile that is associated with those inflorescences or this plant. But those reports are only from southeastern United States. So out there in Guyana, what I did was I just collected observations and took pictures of what insects were coming to visit Xyris, just in general, different species. And then I also focused on our hosts or known hosts, Xyris rudimensys. And for those, I actually have video footage that we collected with GoPros to try to do the insect visitation study. But what we're seeing is that there's a wider variety of insects that come visit. Just in Xyris in general, we actually were able to find pupal cases of that moth on Xyris spike. And we have, a lot of different hymenopterans, coleopterans, and even other arthropods like spiders that we find coming to those inflorescences. And we see the same thing in region 10, a lot of different insects that come visit, even orthopterans, which we were not expecting something that big to come to such a little flower. But we are seeing a lot of different things that just haven't been reported in the literature before. And then looking at our specific known hosts, plant hosts, we actually found that insects come visit both pseudoflowers and the true inflorescences from the plant. And again, it's just a diversity of different insects that come by. Interestingly enough, we also found that some of those insects are actually eating the tissue. So the floral tissue of Xyris, and like that ant just went away carrying a petal. And in this one, we have a cricket that comes by and just like munch on the flower. Now, there are other observations or orthopterans that visit flowers and eat the flowers as supplementary nutrition. Just something that hasn't been reported for Xyris, and that also begs the question whether could they also be eating the pseudoflowers and not just the flower tissue. And then looking at the volatiles that are produced by Xyris and the fungus, the study that described the new species, they did a separate study where they looked at the volatiles produced on pure culture. And they found, they looked at a few things here and there, but nothing that was very conclusive. And we didn't have data for actual pseudoflowers out in the field. So that's what I did. We made this little bouquets of pseudoflowers and inflorescences. We collected from the headspace. And I did the same thing with pure cultures to make comparison. And what we've seen, and this is just part of the results. I know it's a lot of like long, weird names probably, but I just want you to focus on that one. That one compound we actually found on the pure culture and on the pseudoflowers out in the field, but not on the inflorescences. So it's potentially a compound that might be involved in attracting insects to these pseudoflowers out in the field. And we don't really know if it's myrrhine or cubebin based on our data, but both of them have been reported from different plants and even fungi. So just something that we could look into further. Okay. And now we're going to focus on the microbiome information that we have so far. So for this section, I collected samples from both pseudoflowers and inflorescences from both the host or the known host species and a different Xyris species that we have not seen any pseudoflowers on that one. and collected on the 2 different sites in the other Xyris species at the Iwakama forest and 1 site that was in common. And I hadn't mentioned this, but the flowers, the inflorescences only last for a day, and then they fall off, while the pseudoflowers can stay, we don't know for how long, a long time, probably. So I was even curious if we were gonna be able to detect anything on those flowers if they only last for a day. So in some cases, I also collected on like 2 different days, just in case. So collected the samples, in total is 110 samples, brought them back to the lab, extracted DNA using the plant DNA-seq plant minikit. Then we amplified using PCR, and for that we use 3 different regions. So we use ITS for bacteria, 16S for bacteria, ITS for fungi, and we also use F, which I'll explain a little bit more later. But the idea of that one is that it'll give us a better picture of the Fusarium that are present in that system. And here I have to make pause and thank Dr. Terry Bell's lab for all of their help. I could not have done any of this without them. And I really have to thank William King and Ryan Trexler because this is the first and only time I've prepared a library for amphiton sequencing. So they walked me through the whole process and the protocol. So thank you. Then we use Illumina MySeq to sequence the genomics core over here. And we've run some preliminary analysis. So we use data to get to producing the ASD table and the compositional data analysis pipeline for all the downstream analysis. And here I have to do another pause and thank the dog group, the Give a Dog a Bone Award from the Microbiome Center for funding all the sequencing for this project. And a special thanks to Taeyong, because he's the one that has been helping me run all the analysis. So thank you. Okay, so I don't have a full story to tell you yet, but I'm going to show you what I have so far. So, just keep in mind, I'm showing you preliminary data, preliminary results. There's a lot of data in there. So if you notice things when I'm showing the figures that you're like, that's interesting, I know about that, please talk to me later. I need your help. All right, so this is what we're seeing for the 16S. So in bacteria, it's definitely a different community in the inflorescence than there is on the pseudoflower. Now, if we look at the family level, again, it's just clearly with the colors to see they're just different. On the pseudoflowers, we see mostly this family and this community. Well, in inflorescences, we're seeing the ones that are from the same host, mostly Acetobacteriaceae, and the different that is the non-host, we see different families that are more relative abundant. Now, again, a lot of information, and trying to sift through all of this, I asked Taeyong to help me look at the genus level within bacteria. I feel like maybe try to get some more information what was there. So we did. But again, a lot of information. Since I'm more interested usually in the fungal side of things. Something I do notice here is that on the pseudoflowers, we see a lot of the Methylobacterium. And looking into that genus, it's been reported to be an endophyte of plants. It enhances plant growth. It inhibits plant pathogens. It's also been detected on different fungi. It's been shown to have synergistic effects when you co-inoculate with arbuscular and mycorrhizal fungi on growth and nutrient uptake. And it's also in multiple studies associated with different fungal hosts. So what is it doing there? I don't really know, and I don't think we've been looking into that type of question so far. But it's potentially a new avenue for research here. And just like this genus, there's a lot of other data that needs to walk through to try to make sense of how it's coming into play in our system. Okay. So now looking at the fungal side of things for IPS, similar story, completely different thing, which, if we think of what the samples are, one is mostly hyphal tissue, it makes sense that that one will be different. And it is different. So this is looking at different genera that are present in this sample. If we look at the different genera over here, those are usually found associated with different plants. However, we're also getting a lot of unidentified ASDs, which could mean it's just not finding in the database that we're using, and we could do a little bit more work to try and identify those. Or they could potentially be new species, which I wouldn't be surprised since there are no fungal, bacterial studies on any Xyris species. Of course, what you're all asking about is that on the pseudoflower side, everything is ID'd as Fusarium. But we also see Fusarium on those inflorescences, both on the known host and the non-host. However, when we're working with Fusarium, using IPS is not ideal. And even though that's the barcode used for fungi, it's not really that useful for Fusarium. So there are issues with the IPS2 section that usually gives us problem with Fusarium, and also there's not enough variability in IPS for us to discern different species within Fusarium. So actually what we use for Fusarium is tests to be able to identify that sort of barcoding scheme for Fusarium. Test is the translation elongation factor 1 alpha. It's a single copy protein coding gene. And so we have it. Usually what we amplify, we use primers EF1 and EF2. So amplifying that whole section, which is somewhere around 700 different base pairs. And that includes 4 exons and 3 interns. What we did for this project is we used some primers that were developed in 2019 by colleagues and collaborators that are located in this area. So the second and the third exons, and it covers part of those plus that big intron in the middle. And we're hoping that that will help us then be able to identify the different Fusarium that we've seen in the sample. Now, to be able to do that, we of course need a database that we can compare sequences to. And our group, since I think 2004, has had a database called Fusarium ID, which up until the beginning of this year was only available as a blast option online. So people go to the website, blast, and that would help them identify the Fusarium that they have. What we did at the beginning of this year was release a new version of it that includes more information on tests that has representatives from all the different species complexes within Fusarium. And we made the file available for people to download so that they can use it for their own purposes, either set up their own blast search, or they could add other sequences to it from results that they have from their lab. So we're actually trying to use this Fusarium ID database, the test portion of it, to help us identify, assign a taxonomy for our test sequencing. And this is what we're seeing in our sample. Now, I'll start with the positive side of things and mention that, we're being able to identify the species level, the different ASVs that we're detecting. I'll also mention we have a few outgroups included in our database. So, some of those will, things that may not be Fusarium that are in our samples will match to those. Even if they're not actually that species, because that's the closest thing that's in the database. I did check manually like a third of all the ASVs, and the assignments that we have are correct. Now, on the not so good side of things, we're having some issues when we run the analysis. You've probably noticed on this side, the inflorescence, that there are empty areas on our bar, and all of those are things that were not assigned taxonomy. I took the time to go over those and blast them against our database, and it does find a match. So it's not an issue that there is no match on the database. There's something happening when we're running the pipeline that is not assigning the taxonomy for those. And that's where I could use your help. So if you have dealt with this before, or if you have any ideas of what we could be adjusting to help us assign taxonomy to everything, that would be great. Just talk to me. All right. But on the writer side again, if we can get this to work, it'd be pretty cool, because then we can make that database that is now formatted to be able to run on data too, available to other people that can do these types of studies as well. And I think it's interesting in the sense that usually when we think of microbiome studies, we don't get to that level of resolution in terms of taxonomy. We usually have to go genus or higher than that. But what we're trying to do here is to be able to get the species level, and maybe hopefully even farther than that. Also important here is that, so of course the Fusarium that we're detecting here is one thing. It's all Fusarium xyrophilum, so our fungus of interest. But what's even more interesting is that we're actually seeing that fungus also present on the inflorescences of plants that do not have pseudoflowers, so that we thought were not infected. But we're also seeing it on plants that are not hosts, or we thought were not hosts of the fungus. So yeah, that's really interesting for us, but then that means the fungus is probably present as an endophyte, maybe in all Xyris. Could it be in other plants that are in the area too? Just we don't know yet. But now we know that it's not only specific to those 3 hosts that we know of that produce a pseudoflower, but it's actually present on other plants as well. But then for some reason, it can produce pseudoflower on just those 3 that we've known. So in summary, we have now reports of insects that visit Xyris. We have provided a detailed study of the insects that visit Xyris ornamensis specifically. We have found 1 volatile compound that is present on the pseudoflowers and pure cultures with potential to be tested for insect attraction. And we've generated the first study of the bacterial and fungal communities on Xyris. And hopefully we'll be able to provide a database to the community that could be used for identifying Fusarium, specifically using amplicon sequencing. Okay, with that, I have a lot of people to thank. A lot of different agencies that provided funding, including the Give a Dog a Bone Award and the Indigo Ag Fellowship from the Microbiome Center, which have supported my project And a lot of different other institutions that have provided funding for traveling to Guyana and all of the collecting permits. A dissertation committee, a lot of different Penn State collaborators. You saw we have a lot of different things that we're looking at in the system, and I'm of course not an expert in all of them. So really great to have all of those connections within the university and have people willing to help out. And then I also have to thank a lot of other collaborators outside the university, especially the ones that helped me with all of the collecting down in Guyana, and other people that have helped more on the intellectual side, providing information and helping us with technical questions regarding the system. And then I'll also remind you that today, as Taeyong mentioned, we're starting the first dog workshop. It's gonna be once a month, and we're meeting today at 1 p.m. So I'll see you there. And then with that, I'm happy to take questions.
Thank you for a wonderful presentation. We tried to encourage first questions from students, so